all 2023 exams. I'm trying to find paper 3 2024 March, but I cannot find it. If I find it, I'll send it to you immediately. We did solve a lot of exams in the lab, but we want to make a plan. Before the 28th of May, before your paper 3 exam, you will at least solve all 2023 exams, March and June and November. Okay? Now, I want to show you something. This is the website that contains all the exams here. Now, 2023, at least 2023. If you can solve 2023 and 2022 before paper 3, before the upcoming 28th, this would be perfect. Now, the March exam, we already did it all in the lab. I want to remind you, this was the exam that needed 2 pieces of beetroot, and I told you to get beetroot. So we already solved March 2023 in the lab. If you go back at the exam papers in 2023, after March, we have June. We will perform all June exams in the lab also because they are doable, thank God. And we have all the materials. So 31, 32, and 33, we will solve all of them in the lab next week, Sunday and Tuesday, Sunday, Monday, and Tuesday. Good. So we will start and go straight to November 2023. November 2023, there were many variants for paper 3. I don't really know why. 31, 33, there is no 32, 34, 35, 36. So we want to solve all those 4. All those 4, we will solve them together, not in the lab because we don't have all the materials. We'll start with question paper 31, November 2023. So I hope you understand what is our plan. We want to solve all 23 exams before the paper 3 exam on the 28th. March 23 is already done. June 23, we will do it in the lab because we have the material. November 23, we'll solve them together. Okay, are you with me? Do you understand what is the plan?
Yes.
Hello?
Yes.
Okay, so let's start with the November 23. Now, this, if you did all the 2023 exams, this would be very good practice in addition to what we did throughout the year. Let's start with this exam. Any exam, before you start, you have by default to identify your variables, the independent and the dependent. Here you have an investigation where the glucose will be broken down in enzyme by enzymes in yeast cells to give you carbon dioxide and water and ethanol. Okay, so you will investigate the effect of temperature on the production of carbon dioxide gas by yeast cells. Now, Farouk, can you tell me what is the independent variable in this investigation? Farouk?
The temperature.
Yes. Okay. So the moment you read the first step, step number 1, you will decide that, 1 minute, I like red. So let me change it to red. Or you know what? I'm going to get it from my old files so that I can scribble as much as I want. This is past papers, paper 3, years, 23, good. So November, I have it. Okay, let's use this. Yes, so from the moment you read this investigation, you will identify the independent variable, which is the temperature, the independent variable. Okay, you will be given the yeast and the glucose solution and the hydrogen carbonate. Our problem in the lab is that we don't have hydrogen carbonate. That's why we cannot do this one, but we will still solve it. And you will need to maintain samples of a yeast cell suspension at different temperatures, count the number of bubbles of carbon dioxide produced. Now, we did use a water bath in the lab and a thermometer, especially last time. Okay, you will count the number of bubbles. Decide which temperatures you will use for your investigation. These temperatures should be below 65 degrees. Mariam, state the temperatures that you will use in your investigation. This is 2 marks. Hello? Mariam? Hello?
Yes, doctor. Can you hear me?
Where are you? Yes, I can hear you. Can you please help me with this question? They said that you will decide which temperatures you will use for your investigation. These temperatures should be below 65. And 2 marks. What are the temperatures that you will use? Mariam? I can't hear you. Maybe she has something with her. Farouk, can you help
Yeah.
me?
Uh,
Okay. Can you help me? Tell me, what are the temperatures that you will be using? And why is this question out of
because
2
you need
marks?
more than one temperature. So 55, 45, 35, 25, and 50.
Very good. You need at least how many
5.
temperatures? 5. Perfect. So you will write 65 degrees, 55, just like you said. And then these temperatures, they have to decrease by the same amount each time. 55, 45, 35, let's say 1, 2, 3, 4, and 25. So they are 2 marks because you cannot just write one temperature. The independent variable, they have to be 5 different sets of independent variables, and they have to be equally distant. So that is why it's out of 2 marks. 65 minus 10, 55 minus 10, so on. And they have to be at least 5. Miriam, are you with us?
Yes, doctor.
Hello? Okay. And then you will carry out steps 1 to 9. As we said, with every step that you perform, you will make a check mark. And while you are performing your steps, you will be looking for the dependent variable. You already decided that the independent variable will be the different temperatures. You will carry out the steps exactly. Every step you will make a check mark with a pencil so that you don't get confused. And you will be looking for the dependent variable. The dependent variable will be whatever you will measure. You will use the same test tube for the hydrogen carbonate indicator. You will set up this apparatus, okay? And you will start timing and leave the apparatus for 60 seconds. And then look at step 6. After 60 seconds, start counting the number of bubbles produced during the next 60 seconds. So, what will be the independent variable, Farouk? What are you measuring in this investigation?
The dependent. So the number of bubbles produced.
Correct. So from step 6, we will identify our dependent variable, which is the number of bubbles, because like they always do, they will ask us to record a table or to design a table. This table is out of 5 marks. Why out of 5? Because you will not only design the table, but you will put results. Okay. Now, help me design the table, Farouk. How will you design the table?
The independent variable on the left, so the temperature on the left.
Very good. So the temperature, and what else should be next to it here?
The degrees Celsius. So what we're gonna measure
Very good.
the unit.
Okay. And what will be on the right side on the table?
The number of bubbles.
Very good. The number of bubbles, and here we don't have units. Good. Now you will take a mark or 2 for that. And then the remaining 4 marks or the remaining 3 marks will be for the exact results. You have to put results. So we decided that we have 5 temperatures, at least always the dependent variable, they have to be sets of 5. 5 temperatures, 5 pH, always 5. So we said 65. Don't write 65 degrees Celsius. The unit is already in the header, so no units in the body of the table. And 55 and 45 and 35 and 25 and the correct number of bubbles. Okay, it depends on your investigation. Now, Mariam, can you tell me what is the trend? There has to be a correct trend. Even if your results or you got confused in the experiment, you will improvise.
As the temperature increases, the number of bubbles will increase.
Can you both hear me now?
Yes.
Yes, miss.
Can you hear me?
Yes.
Hello?
Yes, we can hear you.
Okay, can you both hear me now?
Yes.
Yes.
Okay, now this connection will be stable. Can you see the screen?
Yes.
Can you see the screen? Okay, Mariam, I was asking you a question. Now, what do you expect the trend to be in case you needed to improvise the results?
As the temperature increases, the number of bubbles will increase.
Yes. Now, I'm not supposed to be telling you that, but a lot of students, they only do the experiment once, to be honest with you. They do it once or two times because you were with me last class in the lab. I don't know, Farouk, if you were there or not, but I think you noticed that maintaining the temperature and creating a water bath is not difficult. It is difficult, I mean. Mariam, weren't you there?
Yes.
Okay, so when we don't have a thermostatically controlled water bath, this electric device, and you will not have it on an exam, you will be requested to maintain the temperature using mixtures of hot water and cold water, because this is the manual method that we use. And this is not easy. Mariam was there and she tried it. And we will do it again next week. I will use an experiment or an investigation that needs us to adjust the temperature because it's not easy. So a lot of students, when they have experiments that involve adjusting temperature, they will not adjust the temperature at 65 and 55 and 45. It will take 3 hours. So I am telling you here as an advice, as a tip, that if you have an experiment on the exam that asks you to adjust the temperature, do one or two maximum. Don't waste time on on the five temperatures. It's going to be very hard. So you will identify the trend, like Mariam said logically, as the temperature decreases, the number of bubbles will increase, or as the temperature, yes, decreases, the number of bubbles will increase, yes. Okay? Mariam said, as the temperature increases, the number of bubbles, what did I say? As the temperature decreases, the number of bubbles will decrease also. So you will do only one investigation at 65, maybe at 55 if you have time, and the rest you will improvise. Okay, so take this tip from me. Only if the investigation requires adjusting temperature. Now state the dependent variable in this investigation, Farouk. Quickly, I want to finish 2 exams, not 1. What's the
The
dependent variable?
number of bubbles.
The number of bubbles. So easy. Observe the final color of the hydrogen carbonate indicator. Now, this is something that you should know, or if you don't, you will have results on the exam. This indicator, when it is exposed to acidic conditions, because we have CO2 produced, what is the color of the indicator? We took it last year in O level. Mariam, do you remember?
Red.
No, it's yellow. Okay, it's okay because you will have an acid and it will be exposed to the hydrogen carbonate indicator. You will do the experiment and it will become yellow. Always this indicator, the hydrogen carbonate indicator, let's revise it together. In average concentrations of carbon dioxide, it is usually orange. Okay, when it is acidic, it is yellow. The hydrogen carbonate indicator, when it is acidic, it's yellow. Remember that. Now, state the final color. We said it. State one conclusion from your observation. Like Mariam said, what is your conclusion? Why does the indicator become yellow? Because the water becomes alkaline or acidic. Excuse me?
Because the solution becomes acidic.
Yes, because the solution becomes acidic. Okay. Now identify one source of error in the procedure and suggest an improvement. This question is a classic question. What is the source of error? You have so many errors in this investigation. We actually did not read it, but what is the source of error?
Counting the bubbles.
Very good. Of course, bubbles can be a source of error. And how can you improve that?
Use a gas syringe to measure the volume of carbon dioxide.
Yes. Mariam, can you think of another error?
We can say the color of the indicator.
Yes, of course. The color of the indicator is always an error. How can you improve that?
Using a colorimeter.
Yes, either using a colorimeter or a white background or a color standard. So this is a very easy question. Paper 3 really is very easy. Now, in paper 3, you will want to practice all the graphs because the graphs are not easy. They're not as easy as they were in O level. Okay. Here you have the concentration of copper ions and the mass of yeast, and you're supposed to plot this on a graph. Now, if you notice, all the independent variables and dependent variables, they follow the same format. 2 decimal places or 1 decimal place. You have to please put this in consideration. Now, what will you plot, Farouk, on the axis? The concentration or the mass of the yeast?
The the concentration, yeah.
Of course, the concentration, and don't forget the units. So the first thing you will do, you will write concentration here and parts per million, the units. And the y-axis will have the mass of the yeast, and don't forget the units. Mass of yeast, okay? And the units. Now, you will put here a 0. But of course, I think you know that you don't always have to start with a 0. You don't have to start with a 0. But how will you distribute the concentration of the copper ions? You have 0.00 and the maximum value is 1. The first thing you will do is that you will divide your graph into boxes. Now, these boxes are obvious. They're clear, thank God. So these are boxes of 10, and this is a 5. Let me divide it into boxes of 5. Okay, we want the maximum value, which is 1.00, to be at the end. How many boxes do we have? Let's say 1, 2, 3, 4, and 5. If we want the 1 to end here, how will we distribute the boxes? Who will tell me? Zero 0.2, 0.4, 0.6. I got it. It's very easy. If I put a 0 here, what will be the value here?
0.2.
Yes, 0.2, 0.2, 0.4, 0.6, 0.8, and 1. Okay, good. And we labeled it. Now, the mass of the yeast, what is the maximum value? 21.2. Let's say 25, because I like it in fives. Okay, how many boxes? This is 1, this is 2, this is 3, this is 4, and this is 5. Okay, 25 divided by 5. So every box of 10 will be how much?
5.
Yeah, this is 5, 10, 15, 20, 25. Okay, can we do a better distribution? This is okay, but this is 21.2. This is very good. This is really good. Or we can do it in 4s if it's going to be up there. So this is perfect, okay? And then you will plot it. Of course, we will not plot it now. And you will plot it either x's or 0 surrounded by a 0 or a dot surrounded by a circle. And then you join with a ruler or a smooth line if it is obviously a curve, and do not extrapolate. Extrapolate means do not extend the line to the 0. There is no value here that says 0, 0. So don't extrapolate. This is incorrect. You will make your graph and then use your graph to estimate the concentration of the copper ions that would result in a mass of 10 grams of yeast cells, and show on your graph how you obtained your answer. So here they tell you, use your graph and show on your graph. So this means that you will go to the graph and you will go to which concentration? No, to which mass?
10 grams.
Yes, you will go to 10 grams on your y-axis. And how do I show on my graph clearly? I will go to 10 grams and clearly make a horizontal line with the ruler until the graph stops me, and then a vertical line and make circle around the value. This will be, for example, 0.4. So you have to show on the graph. Copper ions act as a non-competitive inhibitor for some enzymes. Suggest an explanation for the results show in your graph. Now, we don't have a graph, but what does the non-competitive inhibitor do? Tell me, Farouk.
It binds to the added site, causing the active site to change. So the
Okay.
activity of the enzyme, so it can form an enzyme sulfur complex.
Yes, you're right. Okay. You did talk about the non-competitive inhibitor. It will bind to a different site and it will cause changes to the shape of the active site and it will not bind perfect. You're right. How will this affect the shape of the graph? If the graph looks like this without an enzyme, with a non-competitive inhibitor, Farouk, it
It
will
will be
be below it, B, or above it, C, or how will it look like with a
below,
non-competitive inhibitor?
below.
Yes. Okay. In case they ask you to sketch a graph. So what you said is correct. Good. Yes, another tip. Before paper 3, you will revise the lesson enzymes, cell membranes, and plants. Those 3 lessons. You have to revise them before paper 3. They don't usually include any other lessons. No, also and gas exchange. So you will study plants and you will study cell membrane and you will study, what did I say? Gas exchange. And we said plants and what else? And enzymes. Yes. Okay, so revise those lessons before paper 3. Until the 1930s, most commercial fermenting equipment for growing yeast cells was made of copper. After this time, fermenting equipment was made using stainless steel. Use your graph in figure 1.3 to suggest why stainless steel was used instead of copper. We don't have a graph, but what do you think this is better? Why is the stainless steel better than copper? Will this improve the reaction or reduce the reaction? What do you think, Mariam?
Yes,
Hello?
doctor.
What do you think? Why would they use why would they use stainless steel instead of copper in a reaction? They said that the copper is a non-competitive inhibitor. So what do inhibitors do to the reaction? It's so easy.
They prevent the reaction from occurring.
Yes, they reduce the reaction, they prevent the reaction. So we will not use an inhibitor when we will use this instead. So question 1 was very easy. And if you noticed, question 1 had no dilutions. Not all the questions had dilutions, okay? But it was very easy. Now, question 2. This is a plant stem. Now, you are at home and you want to practice question 2. You can easily go to the internet and you will go and draw. You will write here in your search engine, I want plant stem. You will do this exactly under microscope. Good, because I want to practice paper 3 before the exam. You will get a lot of images. Fine. Let's say you will choose this one. You will magnify it and you will draw it just exactly like we do together. I send you pictures if we are not in the lab and you will draw something like this. Good. They will tell you quickly in the question here, draw this region. Let's draw it together. You should be now experts in drawing. You will not draw any cells. You will draw a very big picture with no broken lines, and you will show the correct proportions. Now, what are the correct proportions? I want to take a snip from this picture and put it next to me so that I draw it with you here. Okay. Where is it? Can I paste it? Paste, paste, paste. Okay, let me go to our Word document. Okay, I'll paste it. Now I can paste it. Now let's draw it together. I'll use black. Okay, now what do I mean by proportions? Because you all have problems with proportions. Help me label this outer layer. What is this outer layer, Mariam?
The epidermis.
Yes, this is the epidermis. Now this is the stem. So do we have an endodermis?
No.
No, we don't have an endodermis photo. The endodermis is always in the root. Now, it's obvious that the epidermis is much thinner, and then we have a layer of cells. These layer of cells, in case you want to know, it's called the cortex, okay? And then we have the vascular bundle, and then we have the pith. but they don't usually ask. Maybe they can ask you to label the cortex. Good. So the epidermis is a very thin layer. I'm going to draw the epidermis like this, thin layer, showing you the different proportions. It's thinner compared to the group of layers that follow it, which are the cortex. Let me make this thicker. Yes. So the cortex will be here. Okay, these are the proportions that they're talking about. This is thinner, and then this is thicker. And then you will draw at least 3 vascular bundles. The vascular bundles are also very obvious. There is a section like this, okay, and then there is a middle section, and then there is another section. It looks like an ice cream cone, okay, and then another one next to it, but obviously this is smaller. Okay, no broken lines. I did not draw any cells, and I can draw one here because I have space. Good. And then the cortex. The cortex is the third layer. This is the cortex. Big, no broken lines, no shading, no individual cells, and hopefully you will take all the 5 lines. Good. So you will draw this here. Now, the xylem cells, you will draw one large xylem vessel element and a group of 3 adjacent smaller xylem vessel elements. Now, you will adjust the specimen under the microscope, and I'm sure now you can do it really well. It's very important that you decide the shape of the xylem vessel. You will draw an empty cell because it's an xylem and there is no nucleus. So don't ever draw a nucleus when they're asking you to draw an xylem vessel element. But why do you need to adjust it under the microscope? Because this xylem vessel element can have different shapes. It can be like this, rectangular, or usually it's not rectangular. Usually it looks like something like this. Okay, it has a hexagonal shape. So you will draw it like we always did, and the only thing you will do is that you will identify or show clearly that there is a thick cell wall and it's empty, no nucleus. And then they said 4. So you will draw 4. And you will not do it like this, no overlapping. Okay, you have to draw a separate cell with separate cell walls for each cell. Good. Now this is horrible. Fine. So why do I have to get it under the microscope? So I know the outline of the xylem. It is rectangular, circular, hexagonal. It's usually like this. But of course, there are so many different shapes for the xylem. And then another one, I want to show you that you will draw separate cell walls for each, because there have to be 3 meeting lines between each cell. Okay. And then a third one here, for example, big, take more than half the space available. This is a very easy question, and you should easily score the mark for that. Okay. Of course, this is horrible. Now, these questions can sometimes be tricky. Here is a micrograph showing the epidermis from another type of plant, and then the actual area of the epidermis visible in the photomicrograph is 0.04. This is the actual area. And the leaf from which this figure is taken has a total surface area of 2000. Use the figure to estimate the total number of stomata on the leaf. Show your working. 3 marks. Now, for what will you do here? Look at the picture. The actual area here is 0.4. What will you do? I will not tell you anything.
Okay. I'll measure the image area.
I'm sorry, Farouk, you will not measure anything.
Okay.
Now, here they're
Sorry.
telling you that.
Okay, so now the total is 2000. And
Okay, that's total.
yeah.
Okay, what will you do? Tell me.
I don't know, to be honest.
Mariam, what will you do?
I'm not sure.
What? You have to think of something. How many stomata do you see?
4.
4. Okay, so if we have 4 stomata, we have 4 stomata in an actual area of 0.04 millimeter squared. How many stomata will you have in a total surface area of 2000 millimeters squared? What will you do?
Cross multiplication.
Yes. Why both of you did not get it? Read the question and understand what they want from you. Here, use the figure to estimate. We will do an estimation to estimate the total number of stomata on the leaf. So they want to know the total number of stomata on the leaf. And they told you that this leaf has a total surface area of 2000. So they want to know the total number of stomata in 2000 millimeter squared. This is what they are asking you to do. And they gave you only one piece of information. They're telling you that the actual area of the epidermis in the figure is 0.04. You will go to the figure, you will count the number of stomata. 1, 2, 3, 4. I have 4 stomata in an area. I have 4 stomata in an area of 0.04. So how many stomata do I have in the total surface area? You will cross multiply 2000 times 4 divided by 0.04. And you will get a number. Okay. Did you get it? Now, these are the questions that are miscellaneous. I call them miscellaneous, and there is nothing you can do about it except practice, because you have a different miscellaneous question per exam. That's why we want to solve as much as possible. Okay? Better? Okay, Mariam. One way to improve the accuracy of the estimate of the total number of stomata on a leaf is to use a photomicrograph with a larger area. State one other way to improve the accuracy of the estimate of the total number of stomata on a leaf. What will you do? Very easy question. Mariam, where did you go? Hello? Can you hear me? Hello? Farouk, can you hear me?
Yes.
What one way will you do to improve the accuracy of the estimate of the total number of stomata? What will you do to improve any investigation?
Repeat and take average.
Yes, you can repeat and take average, or you can choose another sample from another piece of leaf. Okay? Mariam, are you with me?
Doctor, can you hear me now?
Yes, I can hear you better.
Okay.
Okay, now hopefully the last question, they will give you 2 pictures and they will definitely ask you to compare. Now they were nice here and they gave you the table and they told you in the first column, you will need to compare the features and then the features between this figure and another figure. Okay, now you need to choose features between the 2 pictures. What are the features that you will choose? And don't tell me I don't know. You are on the exam and you have to choose differences. What are the differences that you can see?
And 2.3, the stomata is sunken.
Yes, of course. So one of the features will be the stomata. Here the stomata is sunken. Here the stomata is sunken or not sunken.
Not sunken.
Yes, they are on the surface. Good. Something else. What is this?
The waxy, the gold cell.
Yes, correct. And here the guard cell is here. Okay. Again, the guard cell can be a difference. Here, the guard cell is also sunken. Here, it's on the surface. You can compare between the guard cells in terms of size. This looks a little bit bigger to me. And then Farouk said something. He talked about the cuticle, correct?
Yeah.
So what about the cuticle here?
It's thick. The thick waxy
Yes.
cuticle.
And here?
It's thin.
It's thin and I don't even see it. Good. Okay, give me another feature. How many marks is that? This is 4 marks. So we want 4 features.
Yeah.
We said cuticle, we said stomata. You can compare the guard cells. What else? Mariam, can you give the last feature?
The cells that are surrounding the guard cell 2.3, they have more cells, the cortex, and the other one, there is less cells.
Okay, this will also be considered correct. Okay, as long as it's valid. And here the epidermal cells, they look small. And here the epidermal cells, they look a little bit bigger. Good. Let's look at the mark scheme. We did not look at the mark scheme at all, but let's look at the mark scheme now. This is question 2. Question 2. Location of guard cells to matter, good. We talked about the epidermis. Size of the epidermal cells. I think this is what we talked about, the size of the epidermal cells. Cuticle, like Farouk said, any valid point. So what Maryam said is also acceptable. Okay. Now, the problem is that in the case they don't give you a table, you need to know that when you come to compare any two features, the table has to be at least with 3 columns. The first column with the feature. Okay. Now, clear features, epidermis, endodermis, root hair cells present or not, trichomas present or not. Now, I want to also remind you of something. If you have a root, then any protrusions will be a root hair cell. But if you have a leaf, any protrusions will not be a root hair cell. These protrusions can be a trichoma. So don't say that I have a trichoma on a root, and don't say that I have a root hair cell on a leaf. Do you understand me? So if you have a cross section of a leaf and they have protrusions on the leaf, don't say that this is the root hair cell. On a leaf, we don't have a root hair cell. You will see this in the other exams. Good. So in case they don't give you this table, you have to make a table, and the first column will be the features. These features need to be obvious features: epidermis, endodermis, the size of the cell, the outline of the cell, the number of vascular bundle, the shape, the cell wall, the cuticle, something clear. So just the environment where the plant shown in figure 2.3 has grown. 2.3. What is the environment of the plant in 2.3 with the sunken stomata, Mariam?
Yes, doctor.
Yes, 2.3 comes from which environment? This
The
plant comes from which environment?
dry environments.
Yes,
It's an
xerophyte. Correct. Okay. Now, the exam is finished. The exam is finished. Perfect. This was an easy exam. It did not have any dilutions. This question was not easy. Actually, both of you did not get this question. This is, I call it a miscellaneous question. Please go back and look at it and look at the mark scheme in case you want to check something. Okay. The remaining exam, it was very easy, and also identifying that you have 5 independent variables, or 1 independent variable, I mean, but 5 examples for the independent variable. Now, we will solve another exam. 1 exam is not enough. Stay with me, I promise. 20 minutes and I'll let you go. Okay, we solved November 31. Let's solve together November. There is no 32. November 33. Okay, June 23, we will do it in the lab. Okay, here, yeast cells contain enzymes that hydrolyze sucrose into reducing sugars. You have the sucrose, it will be broken down by enzymes in the yeast into reducing sugars. This experiment needs sodium alginate solution, and we don't have it. You will investigate the activity of the enzyme in yeast cells. The yeast cells will be immobilized in sodium alginate beads. You are provided with the material. If any solution comes in contact with your skin, it is recommended that you will investigate the activity of yeast enzymes by using different numbers of beads. Farouk, what is the independent variable?
The number of beads of yeast cells.
Yes, so the number of beads that contains the yeast cells. These are the independent variables. That's the first thing you do. Good. And then you will carry out the investigation. Of course, we will not do it now, but let me open it from my resources. We said November. I don't have it. I download it so that I can scribble. Where is it? This one? Yes. Okay. So we decided the independent variable is the number of beads that contains the yeast. And then we'll carry out the investigation. Of course, each step you will make a check mark so that you don't. Now we are looking for the dependent. After 5 minutes, dip the beads, you will test the activity of the yeast cells by using different number of beads. Okay, okay. Of course, this is not what you will do. You will actually do the experiment. Carry out step 10 to 20, put 16 beads with 10 sucrose solution. Start timing. Okay, start timing and leave for 5 minutes. While you are waiting, set up a water bath ready for step 14. In step 19, you will use a water bath to carry out the test for reducing sugar. State the temperature you will use. Okay, now what is the temperature that you use for a Benedict solution? For a Benedict's test?
70 degrees.
70 or 80. Okay, so above 80, just be on the safe side. 80 degrees and above. Good. You will heat the water bath at the end of 5 minutes. Stir the contents. Use a syringe to transfer, blah, blah, blah. Repeat. Good test tube. Record your results. What is the dependent variable? Oh, yes. Here, look at step 19. If there is no color change after 2 minutes, stop timing and record as more than 120. So what's the dependent variable?
Time.
We are testing for the presence of sugars. The what? What did you say, Mariam?
The time taken.
You sure? No. If there is no color change.
But they did say that we need to record the time.
They did? Okay. Put the test tube in the water bath and time. Oh, yes, you're correct. Thank you. And time how long before the appearance. Good. So let's design a table. Record your results in AII. You will go to AII. Again, this is 5 marks because you have to do the investigation and you have to put results in your table. So what will your table look like? How will your table look like? What is the independent variable we said?
The number of immobilized beads.
Yes, the number of beads. And then what is the dependent variable?
The time taken.
Now, what are the units for the time taken for the color change?
Second.
Yes, correct. Now, another tip. When the time is in seconds, you never give a decimal. It's always a whole number. So the number of beads, at least there should be 5 numbers of beads, 3, 4, 5, and the time. That's why it's out of 5 marks. Now, tell me, what do you think the trend will be? We will assume that the more the beads, the more the enzymes. So the more the beads, it will take more time for the color to change or less time?
Less time.
Correct. Okay. So more beads, in other words, more enzymes. It will take less time for the color to change, and that color to change is a positive result for the Benedict. It means that the sucrose has been broken down into reducing sugars. Now, what is the independent variable? We already said it. It is the number of beads. Good. Now, state one main source of error in this investigation. What is the error?
Judging the color.
Okay, yes, it is correct, of course. The color is very, it is a main source of error. But when surprisingly I looked at the mark scheme, they gave me another source of error and they did not say any valid point and they did not say color can be accepted. So let's say color is correct. Of course, the color is always a correct source of error. But what is another source of error? One main source of error, something else other than the color. Mariam, can you think of another source of error?
The size of the beads may vary.
Yes, of course. The size of the beads will vary. Now let's look at the mark scheme. I want to show you the mark scheme because sometimes they do surprise me. This is, we said this is what? So this is paper 3 mark scheme. We finished 31, mark scheme 33. The error here, see? The source of error, they did not mention anything about the color. They said beads are different sizes. Beads are not all left for the same time in the sucrose. Sodium alginate and yeast may not be mixed. They did not once mention anything about the color. Okay, so write both. In my opinion, write both. Okay, are you with me?
Yes.
Yes.
Okay. Suggest what substances the student put in the beaker for the control experiment. Now, this is a nice question. Here, they used enzymes in this investigation. When we have enzymes or any living tissue in an investigation, what will be the control?
No enzymes.
Very good. No enzyme or boiled enzyme. Good. So put this in mind. Any investigation that requires a plant or an enzyme or a piece of living tissue, the control will be the absence of this living tissue or boiling it. I'll mark those two. Good. So that you go back and look at those on the night of the exam. 2023 exams, all the 4 exams and the June and the May, they give you everything you need before the exam. Describe how would you modify the procedure to investigate the effect of changing the concentration of the sucrose solution on the rate of the hydrolysis of the sucrose. Now, this is a very important question. You will make another investigation, but you will change the independent variable. How will you modify this procedure to investigate the effect of changing the concentration of the sucrose solution on the rate of hydrolysis? Farouk, give me an answer.
Instead of changing the number of the beads, we change the concentration.
Okay, this is 2 marks. How will you change the concentration? And how many concentrations will you have?
5 concentrations
Yes, and how will you get different concentrations of the sucrose?
by adding the sucrose. Not the, sorry, by adding the enzyme into the.
How do you get different concentrations of any solution?
You just said I diluted the water.
Exactly. So why is this 2 marks? This is also a very important question. This exam is harder than variant 31 because the independent variable here will change. We will change the independent variable. The independent variable will be the different concentrations of sucrose. This is not enough as an answer. You have to give at least 5 different concentrations of the sucrose. We know that we change the independent variable and we have to give 5 examples for this independent variable, at least 5. This always needs to be mentioned. Not only that, you need to mention how will you obtain the different concentrations of the sucrose by serial dilution, by proportional dilution, that you will have to mention the term dilutions. I will get different concentrations of sucrose by dilutions. And also, you will mention at least one constant. What will be the constant now? When the independent variable before was the number of beads, now the number of beads will be a constant factor, the number of beads. Okay, this is a very important question. This question is common. And we also answered a question like that last time we were in the lab. You will change the independent variable. It's not enough to say that I will change the independent variable. This question is out of 2 marks because you have to say, how will I change the independent variable? If, for example, temperature, I will use a thermostatically controlled water bath, and I will provide 5 different temperatures. In this case, I will change the concentration of the sucrose by serial dilution or proportional dilution, and I will provide different concentrations of sucrose, at least 5 concentrations. And what was previously the independent variable now will be a constant. Did you understand this question? Now, we have to look at the mark scheme to show you that this is exactly what they wanted. This is the mark scheme here. This is, look at the mark scheme. You will not only say, I will change the concentration of sucrose. You will use a set number of yeast beads. This now became a constant. And what will you change? You will change at least 5 concentrations of the sucrose. And you have to mention how will you change the concentration of sucrose? By proportional or serial dilution. So this answer is very important. This question is common. How do you repeat the investigation with a different independent variable? You will mention how you will obtain this independent variable. You will give 5 examples of this independent variable. And what was previously an independent variable now will become a constant. Is this clear? Maryam, is it clear?
Yes.
Okay, important. This is important. Now, another investigation. Let's go directly to the graph. What will be plotted on the x-axis? Temperature or volume of juice?
Temperature.
Very good, temperature. Now, the maximum temperature here is 80 and it starts with 30. So I'm going to go and write temperature here, degrees Celsius. I'll divide it into boxes of 5 or 10s, whatever. Now, how will I divide this graph? I want the 80 to be close to the end here. Actually, it is designed in a way. How will you do it? Farouk, can you tell me?
Oh, sorry, miss, my internet was lagging. Can you say it?
How will you distribute that?
Can I see the table, please?
Of course, it's 30, 40, 50, 60, 70, 80.
And okay, so 0, 20, 40, 60, 80, 100, and
Okay,
20.
20, 40, 60, 80. Mariam, can you think of something better?
We can do 15.
Yes. Or you can also something better, much easier because it's obvious. You know what I would do? I will here divide this. This will be 30, 60, and then 60 plus 30 is 90. Okay. So every 10 boxes is a 30. This is 0, and then this is 30, and then you add another 30, and then you add another 30. What do you think? Mariam, what would you do?
Um, I would have started the 30. No zero. I started 30
Okay.
from the beginning, and then each 5 boxes is 10, so 30, 40, 50, 60, 70, 80.
Yes, this is also correct. 30, 40, 50, 60, 70, 80. Actually, this is the easiest because the x-axes are 30, 40. This is really easy. So she will not start from a 0. She will start with the 30, and every 5 boxes has a value of 10. And this is also very correct. Okay, Farouk, did you see what Mariam did?
Yes.
Okay, good. Now the y-axis, what is the maximum value here? 98. Let's say 100. Okay, so how will you distribute that on the y-axis? What is the y-axis? The volume of the fruit juice in centimeter cubed. So I'll write the volume slash centimeter cubed. Okay, and then I'll divide it into boxes of 10. How will you divide that, Farouk? I want the 100 to be up there. 2, 40, 60.
Uh,
That's very
each,
easy.
each 10 is 20. Each 10 boxes 20.
Yes, 20, 40, 60, 80, and then 100. Of course, you will plot using X's or 0 inside the 0, and then use the ruler and perfect. Okay. Use your graph. Again, the same question as variant 31. I will go to my graph to estimate the volume of the fruit juice extracted at 55 degrees. I will go to 55 degrees and clearly show that I'm going to use my graph. Let me change color. Okay. I'll go to 55 degrees. I'm going to go move vertically until the graph stops me, and then I'll get a value and show it on the graph. Don't erase it. Describe the change in the volume of the fruit juice shown by your graph. So you need to have a graph so that you can describe the change in the volume of the fruit juice. But can you guess if there is more temperature, there will be more fruit juice or less fruit juice? What do you think?
You miss. Can you repeat? Because it was like.
What do you think if there is more temperature, like as the temperature is increasing, there will be more fruit juice released or less juice released? Here they use the
What?
pectinase enzyme. Yes, okay. And don't forget to give numbers. So as the temperature is increasing, there will be more fruit juice. I did not see the graph, but most probably the temperature when it is too high. Mariam, do you think there will be any juice extraction?
No, because the
No.
enzyme
Okay.
would
Yes. Okay. Now another table here, temperature and the volume, or did we suggest an explanation for why more fruit juice was obtained when using the free pectinase between temperatures of 30 and 50 and when using immobilized. So this is also the lesson enzymes. Why do we use free better than immobilized when there is more fruit juice? What are the advantages of free over immobilized enzymes?
more chances of successful collisions, and the active site is not covered by the beads.
Of course, okay. When they are immobilized, yes, there are advantages. They're more thermostable and they don't denature easily, but they can't move and the active site can be not exposed. So when they're free, they can move freely, more collisions, more exposure of the active site, more enzyme substrate complex, faster reaction. Now, of course, This question can be in so many formats. What are the disadvantages of the free? What are the disadvantages of the immobilized? So go back and study this part and the lesson enzymes. The lesson enzymes in general, it's important before paper 3. Okay. Where are we? Which number is that? Okay, 5. So 1, 5. Suggest an explanation for why more fruit juice was obtained when using immobilized. Now, this is what I was just telling you. Here they used immobilized pectinase between 60 to 70 when using free. So here they changed the question. I was just telling you that. What are the advantages of using immobilized at high temperatures? Farouk, I just gave you this question and I gave you the answer. When you have immobilized enzymes, why do they give you more
Because
results at higher temperature?
because they're more thermostatically stable, they can survive higher temperatures.
Yes, they are. Okay, they are more thermostable. Let me choose. They're more thermostable. Okay, or you can say that the free will denature at low temperatures. Good. Okay, we're done with question 1. No, we didn't. We're not. Oh, yeah, question 2. Now, here, this is the plant leaf. So, again, I'm not in the lab. I don't have a microscope. But if I was in the lab, then what will I do? I will go to my internet and I will write here plant leaf under the microscope. And you will choose, no, this is not what we want. We want cross section. Okay, yeah, something like this. This is a nice one. Okay, and it's clear. And we are supposed to draw it. Now, in a leaf, do we have root hair cells? Do we have
No,
root hair cells in a leaf? Hello?
we don't know.
We don't have a root hair cell. Okay, I cannot paste it here. So we will go paste it here. There's no, where's this? No, I didn't document. Good. I'll paste it here. And then I will draw it. Okay. Very easy. The upper layer is the epidermis, and it is thinner compared to the remaining layers. This is what they mean by proportion. So the upper epidermis like this, the upper epidermis. And then we have what's this layer? And what's this layer? And what's this layer? What's the layer below the upper epidermis, Farouk?
The cortex,
No. This is a leaf.
the palisade.
Yes, the palisade mesophyll. I will not draw cells. I will just draw, and the spongy mesophyll is nearly the same thickness. And the last layer is what, Mariam? Mariam?
Lower epidermis.
Very good. And the vascular bundle is in the center here. Okay. Of course, the exilum is always towards the upper surface. So the exilum is here. These are the exilum here. This is the plum. It's not very clear. Okay. So we'll draw it without any broken lines. Obvious, clear. How many marks is that? Where is my exam? I want to finish. Okay. Now, I will not do this for the 10th time, but here, these are not exilum cells. These are plant cells, 4 adjacent epidermal cells. Again, if you see the nucleus, you will draw the nucleus. You will draw a faint circle, the nucleus. If you don't see it, then don't draw it. In an exilum, never draw a nucleus because it doesn't have a nucleus. Now, epidermal cells, let's say worst case scenario, your microscope, there's something wrong with it and you cannot bring these specimen into focus. Epidermal cells are usually rectangular and next to each other. So epidermal cells usually are next to each other and they're usually rectangular, but not always. Good? Like this. And the cell walls are clear. Good. And use one ruled label line to identify the cell wall. Ruled. So you will use the ruler for not with your hand. You will use the ruler, no arrows, and say cell wall. Okay? Now, this is another leaf. This is a monocot leaf. Now, identify 3 observable differences. Look here. No table. In the first variant, there was a table. But here, there's no table. How will you design the table, Mariam?
We
How
will
many
have a
columns?
column for feature and column for the
Excellent.
2.1 and for K1.
Excellent. So column for features and then column for figure 1, and then column for figure 2. And you will write clear features. This is a leaf. So upper epidermis, lower epidermis, vascular bundle, shape of the bundle, location of the bundle, distribution of the bundle. They love bundles. Good. If there are any trichomas present, not present, fine. But no root hair cell in the leaf. Let's look at the mark scheme and let's look at the features because we don't have. Is this our exam? 1 minute. This is 33. Yes, we're right. Good. So where is the good? So here, number of vascular bundles, size of vascular bundles, size of xylem, epidermal layer, trichomas. Look at the features. They love the location and distribution of the bundles. 3 features, 2 features along the bundle, 1 for the trichoma. If you see any protrusions on a leaf, please don't tell me it's a root hair cell. Root hair cell, my dear, is in the root, not on the leaf. On the leaf, any protrusions, they are trichomas. Good. So the graph should look like this. The first column features, and then the first slide and the second slide, just like Mariam said. Okay, we want to here. This is one of the miscellaneous questions that we're talking about. Measure line P and line Q. Now I want to ask you, when you measure anything, what are the units you use for measurement? Farouk. Hello? Farouk. Mariam, what are the units for the lines?
Micrometers.
No. You will first measure using a ruler.
So centimeter. And then
No.
we multiply by 10
yes.
to get it millimeter.
We multiply by how much?
10.
10. Okay, so you'll get the length of P in millimeters, like Mariam said, and then you will get the length of Q in millimeters. Now, Mariam, calculate the percentage difference in length between the vascular bundle P and the vascular bundle Q, and show your working and give your answer to 2 significant figures. How can you calculate the percentage difference in length? Hello?
We'll subtract the length of vascular bundle P from Q. Then
Correct. So Q minus P, and then?
Over P.
And then the answer? Over P, correct. Over Q. 1 minute. I think it's over Q because Q is larger. Let me look at the mark scheme. And then over Q and then times 100. No, we have to look at the mark scheme. I think we will divide it over Q. Show the length, measure the length of Q, show the length of Q minus the length of P. Okay, we got that. And then divide by the length of Q. Q, Mariam, not P, because it's bigger. Multiplied by 100. Because we want to see how many times P is smaller than Q. Are you with me?
Yes.
And then you will record your answer to 2 significant figures. Farouk, are you there? This is one
Yes.
of the miscellaneous questions. Did you see this question, my dear? This is one miscellaneous question that has to come up in any exam. They wanted the percentage difference, so we subtracted them, and then we divided by the bigger length by Q, and then we multiplied by 100. Okay, Farouk? The exam is finished? Thank God. Okay, Farouk, were you with us in this last question?
Yeah, I was.
Okay, so this exam, also there were no dilutions. Did you notice that? No dilutions in this exam. And this exam was testing the lesson enzymes. So variant 31 and variant 33, there were no dilutions. Good? Okay, enough for today. We solved 2 exams. You can solve so many exams. The problem is with the miscellaneous question. Any questions?
No, thank you.
Okay, I will leave you tomorrow. What is tomorrow? Tomorrow is Saturday? Tomorrow is Saturday?
Yes.
No, let us meet tomorrow. 1 minute. Sunday, I have online whatever. No, we'll meet Sunday also. I'll tell you on the WhatsApp group, because Monday we're going to the lab. Monday we will perform at least 1 June 2023 exam or 2, because we'll go Monday and we'll go Tuesday. On Wednesday they will be doing some renovations in the lab, so we will not be able to go Wednesday. We will go on Sunday, Monday and Tuesday. So Sunday we'll meet. I'll tell you. Okay. Any questions before I go?
No, doctor, thank you.
Okay. You're welcome. Bye.